Okay, so here we are in metabolism, looking at metabolic pathways. So we've already talked about enzyme regulation and the key components of metabolism. And forgive me, I have allergies, so if you hear a stuffy nose, that's what it is. This set of lecture slides is on the central metabolic pathways in catabolism, looking at glycolysis, Krebs cycle, and then aerobic and anaerobic respiration with the electron transport system. And then we'll talk about fermentation and anabolic processes. There are a couple more key concepts for this one. So we already talked about key concepts in enzyme regulation, and here it is aerobic and anaerobic pathways, and I'll draw this out on the board for you as we go along. The key components for glycolysis, Krebs cycle, electron transport chain that you do need to know, and then the concepts of fermentation. Specifically, we'll be writing out energy production for in the form of ATP, reduced electron carriers, NAD and FAD in their reduced form, and any key metabolic intermediates as we go through this process. So what are these central metabolic pathways? Specifically, we're talking about the process of glucose being oxidized through glycolysis and then completely oxidized through the process of Krebs cycle. This has alternate names, the TCA cycle or tricarboxylic acid cycle, citric acid cycle, if you've heard of any of these before in any of your other classes. It can be called all of those things, so we're going to call it the Krebs cycle. The purpose of these processes and why they're coined the central metabolic pathways, the purpose is to form these intermediates. These could be used for substrate level phosphorylation of ATP. In fact, many organisms utilize substrate level phosphorylation. Well, I should say there are times that substrate level phosphorylation is the sole production of ATP. This is, if you recall, where a substrate that has a phosphate group on it can donate the phosphate group directly to ADP to form ATP. This is a very energy inefficient process. Our cells can switch over to doing substrate level phosphorylation to generate ATP energy under essentially under high stress circumstances where oxygen, where our glucose stores have been depleted and our cells need to produce ATP very rapidly. We will switch over to primarily using substrate level phosphorylation. This is otherwise, I guess you could coin this as fermentation. Fermentation is producing ATP through substrate level phosphorylation. We'll talk about the rest of the process shortly, but this is the key production of ATP through fermentation. Secondarily, to oxidize and generate reducing power. This is in the form of these reduced electron carriers, NAD to NADH plus H plus, and FAD to FADH2. And then finally, these intermediates that can function as precursor metabolites. And these precursor metabolites, we've talked about them before, but they can be essentially pulled out of any of these processes and then be put into alternate processes, either fermentation for a very quick production of ATP or even biosynthesis. So if a cell is energy rich and is ready to build more parts of itself, these precursor metabolites can go into biosynthesis processes. So we're looking at catabolic processes specifically with glucose carbohydrate, but we'll talk downstream about using non-carbohydrate substrates. So if you notice here, these are the three key carbohydrate catabolism processes that a cell can undergo. Not every cell can undergo all of these processes. In fact, most can only undergo one or two of these. Very few can undergo all three of these processes to break down carbohydrates to produce energy. But nonetheless, let's just take a look at this. I'll have this in a few different forms for those of you who are more visual. But if you note, there we go. Note that for aerobic respiration, anaerobic respiration, and fermentation, all three of these processes begins with the same pathway, which is glycolysis. And this term you can understand, glyco refers to sugar or glucose, and lysis is to break down. So breaking down sugar or breaking down glucose in particular. Krebs cycle is used to completely oxidize glucose or the remnants of glucose in the respiration cycles, aerobic and anaerobic respiration. Whereas fermentation does not have this Krebs cycle. In fact, glycolysis terminates with pyruvic acid that is then shuttled and converted into an acid or alcohol end product for fermentation. For both aerobic and anaerobic respiration, you'll find that both of these will terminate in the electron transport system. For aerobic respiration, oxygen is the final end electron acceptor. For anaerobic respiration, an oxygen-containing compound will be the final electron acceptor. Again, a different format, same information. You can visually see the similarities between the two respiration processes, aerobic and anaerobic respiration, and note that oxygen really does not come into play until the very, very last step in aerobic respiration here as an end electron acceptor in the electron transport system. Anaerobic respiration, therefore, can still undergo the exact same central metabolic processes, but this one terminates with an oxygen-containing compound, not oxygen itself, something like a sulfate, nitrate, or carbonate. There can be others here. And these ones will, both of these generate ATP through the process of oxidative phosphorylation and a significant amount of ATP through that process. Versus fermentation up here at the top, again, only undergoes this initial metabolic process called glycolysis to generate substrate level phosphorylation of ATP, very small amount of ATP for input of glucose. And then from here, it terminates with an organic alcohol or acid product. Note again, note in particular the energy yield here. So for fermentation, only a net total of 2 ATP can be generated from substrate level phosphorylation for every 1 input of a glucose molecule, versus the respiration processes can either produce max of 38 or 36 ATP, again for every 1 input of glucose. So these are a highly efficient process, and especially considering the way that biology and chemistry works, efficiency kind of rules all. So when we go downstream and look at organisms that can sort of switch over from one process to another, just keep in mind that the most energy efficient process will be preferred by most organisms. So for example, if an organism can undergo both aerobic respiration and fermentation, if provided with oxygen, they will preferentially undergo the fermentation process that produces the most energy yield for every input of glucose. So these guys would preferentially undergo aerobic respiration if possible, and then only fermentation when oxygen has been depleted. Here's one more way to look at these, and this one, it's really just focused on processes that can occur under either aerobic conditions or anaerobic conditions, right? So aerobic organisms must undergo aerobic respiration to survive. Anaerobic organisms can undergo either anaerobic respiration and or fermentation. If you recall, these are the 3 different processes we were writing down from chapter 6 when we were talking about oxygen preference or oxygen requirements, how to classify organisms based on their oxygen use. So let's start with the process of glycolysis. Glycolysis takes glucose and converts it to pyruvic acid or pyruvate as a metabolic intermediate. This process will yield a net total of 2 ATP. 2 ATP are actually used in this process to phosphorylate some of the metabolic intermediates here. 4 will be produced through this process. So we say a net total of 2 ATP, and this is all through substrate level phosphorylation. There are reduced electron carriers generated in this process, specifically 2 NADH plus H+, which is the reduced form of NAD, if you recall, are generated here, and we'll see downstream. These guys have an end destination during respiration processes to donate those electrons and protons to the electron transport system. Glucose is used here as a readily available source of oxidation, so those bonds can be easily broken. Recall this is C6H12O6. So all of those bonds are easily broken through these glycolytic processes and then downstream through Krebs cycle. There are many sources of this, including polysaccharides like we've looked at before with the starch test, specifically testing for the presence of amylase enzyme. For animal cells, we have glycogen that we store in some of our tissue that is our source of, or our storage of glucose that we can then liberate upon need. Alternately, organisms can bring in other types of sugars, including disaccharides like maltose and lactose. We've looked at this already before, that those bonds can be broken between that galactose and glucose to liberate that glucose and use that for respiration. Similarly, for maltose, this disaccharide can be split and converted into glucose. So I think this video is, I know this video is on our Canvas page. I definitely encourage you to go look at that one. I'm not going to click on it here, but you're welcome to watch this process where a 6-carbon glucose molecule is partially oxidized through the process of glycolysis to yield 2 3-carbon pyruvic acid. Let's look real quick at some of the things that your book has for you. Your textbook gives this very brief table, which I don't like very much, but let's look at it. This shows you this initial starting compound, a 6-carbon glucose molecule, C6H12O6. This just to remind you of this concept of group translocation. When glucose is brought into the cell, as it crosses over that cell membrane, it's actually immediately converted to, not this molecule, to a glucose 1-phosphate. So there's a process that glucose is modified in a step-by-step orientation as it kind of moves through the cell. This is showing you this intermediate that's generated from the end of this linear metabolic pathway, fructose 1,6-diphosphate. You'll actually see a phosphate here and a phosphate here on the first carbon, and the 6th carbon doesn't exactly look like this, but nonetheless, this fructose 1,6-diphosphate is then split in half, where you have a 3-carbon monophosphate and a 3-carbon monophosphate here that are then linearly modified with the addition of another phosphate group, and then those 2 phosphates can then phosphorylate ADP to form ATP. So we have an input of 2 phosphates here in this first linear pathway. Once it branches here, there is then substrate level phosphorylation events that can occur here and here. Total 4 ATP are generated in these 2 pathways down here. 2 ATP are used up essentially to donate those initial phosphates. So a net total of 2 ATP are generated in this process. Note that because of this branching that occurs here in glycolysis, then there are 2 equal products of pyruvic acid or pyruvate that are generated at the end of the glycolytic pathway. In addition, during this second step of glycolysis, one here and one here, an NAD is reduced to NADH plus H plus for each of these linear pathways here, which makes 2 total reduced electron carriers generated during this process. So complete, If you are interested, you are welcome to look through this. You do not need to know this information, but just for those of you who want a little bit more detail about where this phosphorylation occurs, this is that initial 3-step process to generate that fructose 1,6-diphosphate. So there's glucose that comes into the cell and is immediately converted to glucose 6-phosphate. It is phosphorylated upon entry, and there's the first ATP that's used up. There are 2 more steps where another phosphorylation event occurs where you have fructose 1,6-diphosphate. So here's those 2 phosphates. This molecule is then split in half during the step of glycolysis to generate these 2 3-carbon compounds that are both phosphorylated. They're slightly different, but nonetheless, during another series of reactions, you'll see an input of a phosphate group to generate a diphosphate 3-carbon molecule. And then both of these phosphates can then be directly donated to an ADP to form ATP. And so note here that this is essentially showing you that for both of these linear pathways from these 2 molecules, essentially the same thing is occurring, where you have a reduction oxidation event during this phosphorylation. So you have a reduced electron carrier generated here, but that's also happening on this side over here. So we have 2 NADH plus H plus generated in this step here because 1 is generated here and 1 is generated here. For the phosphorylation events, here's a substrate level phosphorylation where this phosphate group is directly donated to that ADP to form ATP through substrate level phosphorylation. This step occurs here, and it also occurs over here. So that's why we see two, right? Here's one phosphorylation event, and then downstream we have another phosphorylation event that occurs. And remember, again, there is another directly parallel pathway right here from this other 3-carbon compound to generate 2 ATP in this process. The end result is a pyruvic acid molecule, 2 of them for each of these pathways, and then all of this net energy and reduced electron carrier yield. So if we were to look at this, how many ATP are produced in glycolysis by substrate level phosphorylation, you can go back and count. Now you would have to answer this in 2 ways. You can say gross, how much total ATP is produced in glycolysis through substrate level phosphorylation, and then answer is different from net total. Net means taking into account how much are used, how much are produced net total, right? So answer that in 2 different ways. For the second part, how much ATP are produced, this should say how many, how many ATP are produced in glycolysis, it's a little late here, by oxidative phosphorylation. This one, think about it. Where have we discussed oxidative phosphorylation can occur? These questions I can answer in our discussion session. You can answer this one just by going back in that information. What's the starting molecule in glycolysis? And then what are the products, right? You can look at all of those things or answer them here. So for yield, ATP, again, 2 different ways to think about this. 4 ATP are produced through substrate level phosphorylation during the entire glycolytic process, but 2 are consumed during that those initial 3 steps in that pathway. So therefore, you can say net total, there are 2 gained in the process of glycolysis through substrate level phosphorylation. For reducing power, there are 2 NADs that are reduced. So there are 2 NADH plus H plus. Technically, you should have 2 there. This is your reduced electron carriers. For precursor metabolites, specifically focus on pyruvic acid or pyruvate. Remember that there are 2 of these, so a 3-carbon compound, 2 of them that are generated at the end of glycolysis. But just note, keep it in the very back of your mind, glycolysis intermediates, essentially most of them or all of them, can be pulled out at any time for other processes in the cell. So there are many different metabolic intermediates, but we're just going to focus on pyruvic acid.